Hierbij open this academic ceremony in which Helene Widowski will defend the academic thesis, The Preterm Lungs and Perinatal Stress, Insights into the Role of Aberrant Endogenous Stem Progenitor Cells and Exogenous Cell-Based Treatment. May I invite you to present a summary of your study and the conclusions of your thesis?
Yes, thank you. Dear Pro-Rector, dear members of the Corona, dear colleagues, dear family and friends. Within the coming 15 minutes, I will give an overview on my PhD project entitled The Preterm Lungs and Perinatal Stress, Insights into the Role of Aberrant Endogenous Stem Progenitor Cells and Exogenous Cell-Based Treatment. When we think of pregnancies, we think of a new life, a healthy baby, and a well-going pregnancy for the mother, but also for the families. But unfortunately, not all pregnancies go as planned. And nowadays, more than 1 in 10 women deliver their baby too early. That means before the 37th week of gestation, and it is known as preterm birth. These 11% of women result in 15 million preterm births every year worldwide. And of these 15 million preterm babies, around 70% encounter complications during the delivery or health problems after birth. These health problems can be influenced by various risk factors. One of them is preterm birth itself. Preterm birth can occur at any time point in the course of the pregnancy and thereby interrupts the normal development of the fetus and the fetal organs. Especially the preterm lungs are tremendously affected by preterm birth. And the earlier the baby is born, as the baby on the left, the more underdeveloped the organs are, such as the lungs, so the risk for complications increases, and also the need for neonatal support. The later the baby is born, preterm, as the baby on the right, the better the lungs and other organs are developed, and the lower the risk for complications, and the less additional support is needed. Apart from preterm birth, also other risk factors can influence the severity of complications. Inflammation hereby plays a central role. Inflammation can occur before birth, so during the pregnancy or after birth. Before birth, inflammation is frequently induced by an infection in the uterus, which is caused by bacteria, fungi, or toxins. We show them here as tiny yellow dots. This infection can spread into the amniotic fluid and is in direct contact with the fetus. This infection then induces an inflammatory response in the fetus, the fetal organs, and also in the uterus. And this inflammation in the uterus is known to be a strong inducer of preterm birth. After preterm birth, the inflammatory situation is maintained in the baby because of the application of mechanical ventilation. Mechanical ventilation is a device that helps you breathing by pushing air into your lungs, keeping your lungs open so that they do not collapse. The problem is that the organs, especially the lungs of a preterm baby, are so immature that the baby cannot breathe on its own, so it is dependent on mechanical ventilation. But at the same time, mechanical ventilation induces inflammation and injury in the lungs. The more inflammatory events occur before birth and after birth, the higher the chances to develop chronic lung diseases, such as bronchopulmonary dysplasia. We will refer to it as BPD. BPD is characterized as breathing difficulties of the baby, and it is diagnosed when the baby has to stay on mechanical ventilation for a long time. These long ventilation periods also lead to prolonged hospital stays, which are a great burden for the baby, the families, but also for the healthcare systems. But to understand where these breathing difficulties come from, we will first take a look into the lungs. First, we look at the lung of a healthy baby, and we see that these lungs are already quite well developed. When we take a closer look into the tissue, with the help of a microscope, we see a well-developed network. This network consists of tiny white compartments and lung tissue walls that are colored purple here. During breathing, the air arrives in these white compartments and oxygen is taken up into those tissue walls. The more compartments you have and the thinner the tissue walls are, the better the breathing can occur. Responsible for the development of such a good lung are the stem cells. The stem cells are responsible for building up the different structures in your lung, but also in the other systems. They are the only cells that can actually do so, and therefore they're highly important in the fetal development. When we take a closer look into the lung of a preterm baby with BPD, we see that the lungs are much smaller, they are less developed, and if we take a closer look into the tissue, we see a different picture. We see that the white compartments are very large, there are just a few of them, and the lung tissue is very thick. Only a little amount of air will be able to reach these lungs, and taking up oxygen with these thick walls is very difficult. And this also explains why the baby is dependent on a longer time of mechanical ventilation. As stem cells are so important in the normal development, we were wondering what happened to the stem cells in a lung that develops that abnormally after birth. To address this question, we used a translational sheep model. We used the sheep as a model as the fetal development of sheep resembles the human development very closely, especially with regard to lung development. And in this model, we focused on the effects of inflammation that occurs before birth on the stem cells in the lungs. To induce inflammation, we first mimicked an infection in the uterus by injecting bacteria or bacterial components into the uterus of a pregnant sheep. We will use this yellow syringe as a symbol for inflammation and infection before birth throughout the presentation. In the next step, we mimicked preterm birth by delivering the lamb too early. To be able to differentiate the stem cells in the lung from the rest of the tissue, we stained them with, in this case, a black color. And as you can see, here we look at a healthy preterm lamb lung. We see a lot of black dots present in the lung tissue, which means that a lot of stem cells are present. When we take a look at the lung of a lamb that has been exposed to inflammation before birth, we see that the amount of stem cells is much lower. And that shows us that inflammation before birth has a strong impact on the fetal development. And we further assume that this lower amount of stem cells might also be responsible for the adverse development of the lung that we see after birth. Specifically, we were wondering if the amount of stem cells was also changed after birth, and we wanted to know if the changes that we see before birth might also impact the lung function after birth. To do so, we extended our model by the addition of another inflammatory event after birth, and hereby we used mechanical ventilation, as preterm babies clinically are also dependent on mechanical ventilation after birth. First, we look into we look into the stem cells. In animals that have been exposed to inflammation before birth with an infection and inflammation after birth because of mechanical ventilation, we see that the amount of stem cells did not change. Although at this point, we still need to look into the function of these stem cells, but this is a topic for future research. Secondly, we looked at the effects of inflammation before birth and its impact on lung function after birth. For this, we looked at the amount of oxygen that is needed during breathing. A healthy person usually breathes the amount of oxygen that is present in the air. That is 21%. However, from the clinical situation of preterm babies, we know that they have respiratory problems and they need a higher concentration of oxygen to supply the body sufficiently. This is usually supplied by mechanical ventilation. In this graph, we will look at the amount of oxygen that the lambs needed in a ventilation period of 3 days. The green dotted line is our reference. It shows the 21% that is present in the air. In our control animals, these are animals that only received ventilation after birth. We see that a respiratory problem is developing. These animals have an oxygen demand. As you see, they cannot stay constantly at the 21%, but it rises up to 25–30%. Which we then supplied with the help of mechanical ventilation. However, in animals that have also experienced inflammation before birth and then after birth, we see that this respiratory problem is much stronger. These animals, and this is the red line that you see, these animals had really difficulties getting oxygen into the body with an oxygen demand rising to almost 60%. Then we were wondering where this bad lung function might come from. And one explanation is that the accumulation of fluid in the lung tissue, which is known as edema. But how does edema develop? Usually the cells in your body, they're sitting very close to each other, and they are tightened together by cell connections. We show them here in purple. These cell connections then avoid that fluid can leak into your tissue. However, when something goes wrong and the cell connections are not working properly anymore or are lost, maybe because of inflammation, the cells are moving away from each other and fluid can leak into the tissue. And this fluid, the force of the fluid then makes your tissue swell and look much thicker. Interestingly, in the animals that were exposed to inflammation before birth and after birth, we saw that the amount of cell connections was much lower than in animals than in control animals that have been exposed to ventilation after birth. We also saw in the tissue that the tissue looked thicker and fluid was present. In a final approach, we wanted to know if we can prevent the changes that we caused with inflammation in these lambs, or at least improve the outcomes that we see postnatally. And therefore, we think a therapeutic component should have strong regenerative capacities to resume proper growth. We find these characteristics in stem cells. But these stem cells are different from the ones that we just looked at. These stem cells are from human origin. They have been isolated from the bone marrow of a healthy donor. They have been purified and prepared for treatment in diseased patients. To test stem cell therapy in our animals, we extended our... Sorry?
Can you close?
Yeah, sure. I'm almost about. We extended our model by injecting the stem cells intravenously. And what we saw is that in animals with an oxygen demand, the oxygen demand was completely gone with stem cell treatment. These animals were able to breathe at 21% most of the time. and we saw that in animals that an edema had developed, that the loss of cell connections was completely prevented in these animals, and also less fluid was present in the lungs. This leads us to the conclusion that inflammation before birth can change the stem cells in the lungs before birth, and it can also impact the lung function after birth. With stem cell treatment, though, we were able to prevent this bad lung function after birth, possibly by the reduction of edema. These findings are clinically highly relevant as they deliver valuable insights into the development of the lungs and propose a new therapeutic option with great potential to help preterm infants that have experienced various inflammatory response before and after birth. Thank you for your attention. I'll give the word back to the prorector.
Thank you for your presentation. The opposition will be opened by Professor Dompeling, Professor Pediatric Pulmonology at this university, and he was the chairman of the assessment committee.
Thank you, Prorector. Dear candidates, I want to congratulate you with your very nice work. I think it's a very nice piece of work with a lot of studies and a lot of nice results, and also I think promising results, because it's maybe possible that these approaches can be also applied in human situations, so that's also one part of my question. So I think very good work, and also a very nice introduction, I think, so compliment with that. And also to, of course, congratulations for your team of supervisors, which also did a great job, I think. My questions has to do with 3 issues. I first want to discuss some methodological issues with you, then about the side effects of stem cell therapy, and the third topic is about planning a human trial and what kind of endpoints are then possible, or what kind of design would you suggest. So let's start with the methodological question. And it strikes me that in chapter 2 and also in the other chapters, you used ureaplasma infection as a kind of chronic inflammation trigger, whereas you compared that with acute inflammation induced by LPS. I was wondering, is it not like comparing apple with pears? You're doing one intervention with LPS and LPS, and the chronic inflammation is more about a kind of microbial stimulus. So can we compare that? Should you have chosen a different approach in future studies? What is your opinion about that?
Highly esteemed opponent, thank you for your compliments, your kind words, and your questions. So we used different inflammatory. Let me start again. The aim of this study was to test various inflammatory events that can occur before birth. With the use of LPS, we also show we do not only focus on the use of an microbial trigger, but also on the fact that an acute inflammatory response can be induced in these animals, which can also be caused by other factors, or delivery itself is also a very acute inflammatory situation prenatally. That is, first of all, the reason why we chose to use LPS.
Yeah, thank you. So, in fact, we both agree that you cannot completely compare it, and I understand your choice, of course. So then a question about statistics. It strikes me a little bit that you don't present much median values or mean values or 95% confidence limits or interquartile ranges. So my first question was, did you check for normality of the data? Did you analyze it by student t-test, or did you use non-parametric tests? Can you briefly comment on that?
Yeah, so we used a non-parametric ANOVA. We did this because the amount of animals that we use per group is a little bit lower because we are working with a large animal study. And therefore, we assume that the distribution of our results is not even normally distributed, and therefore we use a non-parametric ANOVA.
I'm happy to hear that. Thank you for that. Then I skip my second question about the possible side effects of stem cell therapy because of time. And then I move on to my last topic. It's about when you should apply these stem cell therapy in premature babies. What kind of, and you have, let's say you have unlimited resources, you are the principal investigator. What kind of design would you choose? Do you think you should choose repeated injections of stem cells or only one time? What kind of important endpoints would you choose for such a trial? And what about side effects?
Yes, indeed. Just to briefly address the last question with the side effects, I think this can best be tackled if we perform longitudinal studies in which we look at in a long-term manner if any side effects might occur. I think that if I would be able to do that, I would choose to, first of all, well, if we look at our results, we see this very beneficial effects on lung function after birth. And also in the preterm brain, we have seen very beneficial effects with this stem cell treatment. And I think that, first of all, we would need more confirmatory studies before we can implement this stem cell treatment into the clinics. And then, of course, there is a possibility that we use stem cells as an adjuvant treatment with other therapeutics together, because there's already standard treatment for preterm infants that we should also take into account. But at the same time, indeed, double treatment is also.
Yeah.
Yes.
Thank you for your answer. I'm satisfied, and give the word back to the prorector. Thank you.
Thank you.
Thank you. The opposition will be continued by Professor Morty, Professor Translational Pulmonology at Heidelberg University. Professor Morty was also a member of the assessment committee, and we highly appreciate your presence during this academic ceremony.
Thank you, Prorector, and dear candidates. So thank you very much for a very nice thesis and a very nice presentation. I think that you've touched on a topic which has given a lot of us working in prenatal care much hope, since we need to find new ways to do better for affected infants. I will confine my questions to two broad areas. One relates to extrapulmonary consequences of preterm birth, and the second one relates to the translational pipeline. So if I turn to my first topic, as you probably appreciate very much, bronchopulmonary dysplasia and other pathologies associated with preterm birth are more recognized now as syndromes, not individual diseases, where you have a component that impacts the respiratory system, but then there are many other organs and systems affected: the brain, the gut, the heart, the liver, the kidney, the eyes, etc. I'm wondering if you have considered this in your studies. I know that you did not present data in it, but nevertheless it is an important consideration whether exogenous stem cell therapy, where we first think how can we repair or protect the lung and inflammation that impacts the lung, but might those stem cells also be doing positive or negative things to the other organs that are affected by preterm birth and oxygen support?
Highly esteemed opponent, thank you for your kind words and your questions. Just to briefly repeat, you were wondering if the stem cell treatment would also have beneficial or in general effects also on the other organs apart from the preterm lungs. Mm-hmm. Indeed, we have shown also in the preterm brain that the anti-inflammatory response was increased by the use of these stem cells. It has also been shown or assumed that macrophages in the brain, microglia, have been polarized into a more M type, so anti-inflammatory kind of macrophage, and that the blood-brain barrier could be protected by the increase of annexin A1. So we do see, apart from the lungs, where we also have very beneficial effects with regard to the improved lung function and the prevention of cell connection loss, that several organs are affected. And I think that with administering the stem cells intravenously, as we do in our model, we have to further look into that. But I think that this intravenous administration is responsible for these effects, as we first of all tackle the systemic inflammatory response that is in the fetus, and at the same time also then locally, leading to, at this point, beneficial effects in the brain, but also in the lung, and hopefully also in other organ systems, which we will see.
Okay, thank you for your answer. I am satisfied with your answer.
Thank you.
As a second question, I would like to highlight the translational pipeline. What I mean by that is many cell culture and other in vitro studies might give rise to an idea which is then tested in a small animal model, perhaps a mouse or a rat, and then this is translated further into large animal models and hopefully develops into something useful that can be tested in a clinical trial. But there's been a lot of disparity comparing what comes out of these different studies. So for example, in mouse and rat models, exogenous stem cell therapy can cure pretty much any lung disease. But when you move into the clinical arena, particularly the studies that have come out now for over a decade from South Korea, you see that there's been much less success in the use of exogenous stem cell therapy in a clinical setting. And I was wondering if you might comment on why this may be the case. And I would like to acknowledge and compliment you on the use of a large animal model. I would like to see large animal models more widely used in these studies because I believe the data there are more meaningful. However, particularly comparing and contrasting small animal models, large animal models, and the clinical situation, you might want to consider different inflammatory signatures. You might want to consider the different structure of the respiratory elements where exogenous stem cells may have different effects. Perhaps you can highlight for us briefly why you think we see such different things comparing the preclinical and the clinical studies, and how we may overcome these in the future.
Yes, that is a very, very interesting question. I think that one of the reasons why we see this difference between the preclinical and the clinical situation is that the clinical situation is very complex in the babies. They have BPD, they have also other diseases. BPD and other fetal diseases have been caused by various inflammatory factors. In a preclinical model, in rodents and in sheep, we are at this point not able to mimic all these different events, especially as they're complex and they're very individual to every baby on the NICU. And I think this is one of the reasons. So we are tackling with the research that we and others do certain aspects, but at this point, yeah, the situation of BPD is very complex still. And I think with our study, we are coming a little bit closer to this as, for example, in rodent models, they often focus on postnatal insults. We, in our study, already combining the prenatal and the postnatal inflammatory events combined, which are very important, as we have also seen in our project that prenatal inflammation has a strong impact. And this is one aspect that is not often combined or taken into account so far.
Okay. Thank you for your answers. I am satisfied with them, and I return the chair to the prorector.
Thank you.
Thank you. The opposition will be continued by Professor Goossens, Professor Translational Pharmacology at Groningen University. Also for you, thank you for your participation.
Thank you, Mr. Prorector, and dear candidate, it's a pleasure for me to be here as well, and I'd like to congratulate you on your thesis and involve your supervisory team in these congratulations. And the questions that I have actually relate a little bit to the previous discussed topic, so the stem cells. I'd like to elaborate on that a little bit more, but maybe just from a, let's say, pharmacological point of view. And the first question I have is related to the choice for this particular stem cell preparation. There's many different choices out there. Local tissue stem cells, bone marrow, mesenchymal versus epithelial. And I'd like to discuss with you a little bit your motivation for using this particular population, stem cell population, for your studies.
Highly esteemed opponent, thank you for your compliments and for your question. Yes, so we have chosen to use multipotent adult progenitor cells. I will refer to them as MAPC. They are also bone marrow derived. There's other mesenchymal stromal cells as well. However, they are a sort of precursor cell. That means they are, first of all, smaller in size, meaning that the biodistribution in an organism is higher with these kind of cells because they do not get so easily stuck in the capillary system of the lungs, and they have been shown to also migrate further to the spleen, the brain, and other organs. More importantly, though, is their strong regenerative and anti-inflammatory characteristics, which we were looking for in a therapeutic because of these many inflammatory situations that we see in the preterm baby and that we also mimic in our model. And for us, it was important that these characteristics are fulfilled.
Right. And so sorry to interrupt you, but just to keep the dialogue going. But is it then morely a mesenchymal or a hematopoietic stem cell? What you're using here? Is it, you say omnipotent, or what do you mean specifically by that?
Sorry, I did not completely.
But is the stem? So it's bone marrow derived, but is it then hematopoietic stem cells, or are they mesenchymal stem cells primarily? Can you elaborate on that a little bit more, or do you know?
I'm not completely sure, but I think they're mesenchymal stem cells because they also have they also express certain cell surface markers that also mesenchymal stem cells, and also other MSCs that are used for therapy are used.
Okay.
They are also
plastic. And it makes sense in the context of these other studies. So but then the question, so the anti-inflammatory, so the immunomodulatory data, let's put it that way, are convincing. I guess you were disappointed not to see sort of strong effects on type 2 cell markers. Maybe you would have hoped that. I wondered if you had considered the discovery of other types of progenitor populations, particularly the recent discoveries in human airways where we have additional stem cell populations that aren't there in the mouse. Whether you would expect maybe changes in those subpopulations and whether they are actually there in sheep or not. I don't have a clue actually.
Of course, we have we have considered to looking at different stem and progenitor populations. And this is also what we want to do, or what we are aiming for in the future. At this point in our projects, we were focusing on a cell marker with which we could first cover all stem cells from one specific population. So P63 basal cells, TTF1 club cells, and type 2 cells. But indeed, it would be very interesting to see how different subpopulations of these stem cells and even progeny cells would be affected by inflammation.
And could you think of ways to test that? Perhaps not involving, perhaps not directly going to the sheep model, but are there ways to also use the sheep or other models to test the efficacy of your stem cell population against sort of specific subpopulations of cells?
Yeah, indeed. I think
in vitro models, for example.
Sorry?
In vitro models, for example.
Yeah, I was just about to refer to the 3D lung organoid models that I think are a good choice to study specific responses of stem cells, because with the growth of stem cells we can look at the differentiation potential, we can look at proliferation and in general changes that the stem cells or intracellular occur in the stem cells when exposed to different stimuli. So I think, organoids are important here.
That might be a food for thought for a new PhD student in that respect. Well, thank you very much for the discussion, and happy to give back the word to the prorector.
The opposition will be continued by Professor Sluimer, Professor Vascular Pathology at Maastricht University, Professor Sluimer was member of the assessment committee, and she is the secretary of the degree committee.
Thank you, Mr. Pro-Rector. Also, of course, my congratulations with the thesis, the design, the content, and also to your supervisory team. And great that despite all the troubles during COVID, you were still able to do these large ML experiments and work with the tissues that were generated and got some really nice insights. So I would like to ask one of the panelists to read a proposition number 4.
Early identification of prenatal disturbances and early treatment strategies thereof in premature infants reduce postnatal injury in these infants and, hence, provide them with a better start into the future.
Thank you very much. So my question actually arises from that proposition, but also when I was reading the thesis earlier this year, in page 19, you were also talking about early identification. But then when I went through the thesis, I was a bit surprised that I didn't find anything about biomarkers or how to identify then people, or in this case, the sheep that were at risk. So I was wondering if we can talk a little bit more about this, and maybe you can tell me how is prenatal infection at the moment being diagnosed in the clinic?
Highly esteemed opponent, thank you very much for your kind words, your compliments, and your question. So, at the moment, infections are diagnosed clinically. Let me start like this. An infection in the uterus is clinically known as chorioamnionitis. During the pregnancy, we call chorioamnionitis the clinical form of chorioamnionitis. It is usually diagnosed due to clinical symptoms of the mother. So there's a uterine tenderness, fever, and other symptoms that we can identify from a maternal point of view. If there is an indication for an infection, it is also possible to perform amniocentesis to take a sample of the amniotic fluid to check for an infection. And the difficulty at this point clinically still is that sometimes you don't have these clinical symptoms, and then you have a silent infection that you only know about once the baby is born, and you have a histologic examination of the uterus. And therefore, it is very important to further look into options how to identify an infection prenatally. And we have our group, but also other groups are already working on some of these methods. And one of them is the use of volatile organic compounds, so that we might be able to analyze the breath of the mother, in this case the ewe, for specific compounds that might have already been changed before any signs can actually occur, and that might also indicate and give us an indication for
okay, well, that's very interesting work as well. And is there a reason why your group has chosen then the breath over the plasma biomarkers that are more commonly used in my field, in the cardiovascular field?
Well, we are of course also looking at biomarkers. We and others, of course, looking in biomarkers also in blood samples. I think the reason, therefore, is also that it is a very non-invasive way of examining, not only in the mother, but also, for example, also in the fetal situation, when the baby is born, that you try to be as less invasive as possible. And I think, therefore, volatile VOCs are very important. And
do you think, if you think about categories of mediators, would you then guess that there will be more inflammatory ones, or also more fibrotic mediators or products, or maybe another category?
I think that definitely also inflammatory, pro-inflammatory, but also metabolic, I think you can also have a look.
Okay, and my last question, have you been able to see any of those changes of your colleagues also in your sheep model? So the things that are being upregulated or exhaled more often?
To be honest, I'm not sure if we are that far already to compare these. I know that we are setting up, I know that these experiments are running, and we will definitely do this comparison then afterwards, but at this point, I don't know if we have already done that.
Okay. Thank you very much, and I give the word back to the pro-record.
The opposition will be continued by Dr. Langen, Associate Professor at the Department of Respiratory Medicine. Dr. Langen was also member of the Assessment Committee.
Thank you, Mr. Prorector. First of all, also my congratulations, of course, to you, dear candidate, with the completion of this, I think, very nice piece of work. Congratulations, of course, also apply to your supervisory team. And my compliments on the very clear and focused description of your data and the way you introduced them in your thesis. So thank you for that. My opposition, I would like to focus on two issues, 2 points and questions actually that were raised. The first one regarding the lung function measurements in chapter 2 and 3, and the second theme would revolve around the way that you describe function of stem cells. With respect to the first question, when I read your data in chapter 2, there is a figure in there that shows that there is an increase, what you refer to as lung function in the sheep that, or the lambs that had undergone the perinatal inflammation protocol. And considering the impact of the the perinatal inflammation that you and your group already has described, you would expect that lung function not to be increased. At least that was my expectation. So I was quite surprised about that. So my first question to you would be to briefly elaborate on that data and maybe explain the underlying biological mechanism that might be responsible for this, in my opinion, unexpected increase in lung function.
Esteemed opponent, thank you for your compliments and your question. If I understand correctly, you're referring to the increase of the lung function data that we see with the administration of or exposure to UP and LPS. Is that correct?
Yes, the 7 days LPS and the UP.
So I think
maybe I can clarify my question. It relates to, I think it is figure 5 on page 55. And there is an increase in what you refer to as lung function. And to me, it was sort of surprising to see that. And I could not really find a good explanation about this unexpected increase. So that's why I would like to know from you, can you explain it here?
So from the clinical situation of preterm babies, we know that the baby is born preterm, that is born preterm and that has been exposed to inflammation before birth. That inflammation, such as chorioamnionitis, is responsible for the maturation of the lung as well. Not only, but this is one part of it. And I think this is also one of the effects that we see here after the exposure to UP, so chronic inflammation and LPS, acute inflammation, that there is an maturational effect that we see because we see, and one of the explanations for this might also be the increase in surfactant, especially in surfactant protein C. We see that after 7 days LPS exposure, which is also the highest increase in those lung gas volumes, this might also be responsible for this, might be the maturational effect.
So it's like an accelerated maturation that would be a plausible explanation according to you for these observations. To what extent are you sure about the way that lung function, because that's what is referred to here, is measured appropriately? Because if I understand correctly, lung gas volume was determined and it was then divided by body weight. To what extent do you think that that approach may also have influenced the outcome and, again, in my opinion, an expected increase in lung function that way?
Well, we have, these data have been normalized to lung weight,
Exactly,
body weight, indeed. And we see that, well, first of all, we did look at body weight and lung weight, and we see between the groups that there was no significant increase in body weight or lung weight, and therefore we do not think that this, also because we correlated for it, that might have an impact on the...
Yeah, exactly. That's what I was thinking about. That maybe the inflammation has a systemic impact resulting in a decreased total body weight, and you get an artificial increase in this measure. But I'm very glad to hear that you ruled out this, well, let's say this potentially inappropriate interpretation, and I do appreciate your biological explanation. I see that we're almost out of time, but final question would relate to stem cell function. You basically divide up function into regenerative potential and immune modulatory potential. And maybe I would only focus on the last part. What do you really mean with immune modulatory potential, and how would you measure it? Because in my opinion, only looking at markers and number of cells in a static way that was performed here would probably not be sufficient to conclude anything about the potential of these cells to apply immune modulatory responses. Do you have any ideas, maybe a short experiment that you could propose to test that?
I'm not completely... You're still referring to the same data set, right?
Well, let me clarify briefly then. I think in some instances you have shown, for instance, in the final chapter, that there was an impact of stem cell treatment on immune cell markers, or not cell markers, but immune markers. And there was actually even more inflammation. My question would be, how would you really study immune modulatory potential, maybe in an ex vivo setting, that could truly tell you something about to what extent immune cells are modulated by stem cells? I think that's what I would like you to think about.
I think that it's, as you already say, out of an vivo study, I think that a co-culture model might be a good choice if we want to test the effects of two cell types. So in this case, the MSCs on the or MAPCs on the immune cells, and I think that the transwell system in which we can grow them together would help to identify or to look at the responses and the reactions specifically. Yeah,
that seems to me like a very viable approach and maybe a good suggestion for your successors. So I'm very satisfied with your answers, and I'd like to return to the word to the prorector.
Thank you.
Thank you. The opposition will be continued by Dr. Lapointe, Associate Professor Cell Therapies for Regenerative Medicine at this university.
Thank you, Mr. Prorector, and dear candidate. First of all, my congratulations to you and also to your supervision team for this very interesting, also very important thesis. I enjoyed reading it. I wanted to begin by asking some questions actually about the progenitor / stem cells in chapter 2 to start. So thinking about like figure 2, for example, where you reported a reduction in the number of P63 positive cells in the proximal airways after the LPS hit. These tissue-resident stem cells, though, didn't just float away. And I'm wondering what you think actually happened to them. Did they differentiate, for example? Did they de-differentiate? What happened to the P63 positive cells?
Esteemed opponent, thank you for your compliments in your question. Yes, indeed, we see that prenatal inflammation after 7 days caused a decrease in the P63 positive stem cell population. I think, as you already mentioned, there are different possibilities. I think it is possible that they have differentiated already either in a progeny cell like the ciliated cell or club cell, or they might have, if I remember correctly, there were also studies where it was shown that they're still keeping their progenitor mode, but they lose the expression of P63. So they're already halfway de-differentiating into different kind. In other studies, it has also been shown that P63, in the case of injury in the lungs, more in the periphery, are able to migrate to a location of injury. And as we have seen, that inflammation is also present in the lung and also in other regions where also stem cells have been depleted. That might also be maybe a way that they have just moved away from the proximal airways, which are in this case a little bit less affected, as we see this milder decrease in stem cells, which might explain the lower number.
Yeah, I think that's a reasonable hypothesis, of course. And then I also have the same question, though, because a similar finding was in the next figure, also with the club cells. Would you expect the same type of maybe differentiation or migration to be happening there, or do you have a different idea for that population?
For the club cells, we also know that different variant club cell populations exist. And it is possible that one of them has been more depleted than the other one. Of course, these have been shown in rodents, and I think it would be super interesting to look in the sheep as well, which of these populations are actually affected, or have they also differentiated into other cells. We also know that club cells that are very at the alveolar junction, that they also are able to form alveolar type 2 cells. So if there was a migrational or sort of change in the differentiation to the one or the other tissue type, that might also be possible.
Okay, yeah, makes sense. Then it's a question then about your experimental choice actually that followed, which actually you're kind of already touching upon. In the next part of this chapter, you looked for whether the cells were still proliferating or whether there was evidence of cell death. But actually you didn't look for differentiation or for other markers. Can you describe a bit and justify that choice?
You are referring to the type 1 data with the aquaporin 5 that we used.
Also the Ki67 caspase studies, so generally those choices of markers.
Oh, the choices of markers, yeah. We saw that, first of all, we see that our stem cells, so the TTF1 positive type 2 cells, that they are depleted. Then we were wondering, indeed, where is this lower number coming from? And looking at function, we then decided to look for proliferation and cell death at the same time. We didn't find proof for cell death, but we see that proliferation is decreased. And then we were wondering, but what happened to these cells if there are less of them and less proliferation is happening? Might that be because of a increased differentiation potential to have more type 1 cells, as these are the important units in the oxygenation in the lungs? So there was the choice then to also look into the differentiation potential to explain where this lower number might come from.
Okay, thank you very much. I'm satisfied with those answers. Back to Mr. Prorector.
Thank you.
The opposition will be continued by Dr. Simons, chest physician at the Maastricht University Medical Center.
Thank you, Mr. Prorector, and dear candidate. First of all, also my congratulations for this thesis and also to your supervising team. I learned a lot. I'm an adult respiratory physician, so I learned a lot about early life stressors as a risk factor for respiratory diseases. Your topic was BPD. I also read that chorioamnionitis increased the risk of asthma, and I wondered if you could help me understand how chorioamnionitis would increase the risk of asthma based on the results you present. Maybe it's a bit overstretching your result, but maybe we could hypothesize on that. So could you help me understand why I would see more asthma patients with children preterm and having had chorioamnionitis?
Esteemed opponent, thank you for your compliments and your question. With regard, first of all, chorioamnionitis clinically is known to be a strong inducer of adverse lung development or contributor to adverse lung development after birth. Our chapters 2 and 3, and where we see that prenatal inflammation reduces the amount of stem cells, further supports this dogma that inflammation before birth has an impact afterwards. The question here is, so we see that prenatally the amount of stem cells is depleted. And we are wondering, and this is also something for future research, but might also explain or be the answer to your question, is what happens to these stem cells after birth? So if they're so important in lung development and if they're depleted, how long does this, what is the impact of these stem cell changes also after birth? And I think, and there has been some association also for COPD, that the changes that we see prenatally might also then have such prolonged effects also after birth into adulthood. And I think that asthma in between might also be affected by this.
And would you think that also the maturity that you talked about there could be an anatomical change that predisposes those children to asthma as well? Because now you're talking about stem cells, but could there be some anatomical changes in these children that could increase the risk of asthma? I just wondered.
Could you elaborate?
So we know the resistance, so the changes during your childhood, and I wondered if you talked about the maturity of the lung in utero, that might also affect postnatal changes in resistance as well, in growth, and thereby in resistance, and thereby increase the risk of asthma.
Yes, I think so. We know that this inflammatory response, induces maturation, but at the same time at the expense of development. So we see temporarily after birth that lung function is improved, but then afterwards the further respiratory problems develop also because of developmental arrest that has occurred in contrast.
You can't finish your answer.
So that the that this developmental problems in comparison with maturation have occurred afterwards.
Thank you so much. The word back to the product.
Helene Widowski, the time appointed for defending your thesis has passed. The degree committee will now withdraw to discuss the quality of your thesis and your defense. I request that you and your company await the results of our deliberations and our return in this room.
The PhD defense has now ended. The degree committee will debate the candidate's performance behind closed doors. This process usually takes about 10 minutes. In the meantime, let's take a closer look at Maastricht University's renowned teaching method, problem-based learning. Once the pro-rector reconvenes the session, we'll tune in and continue the live stream.
Problem-based learning, or PBL. What does that mean exactly? 3 of my fellow students and I will show you around. Every week, we analyze a different case or issue together. We discuss the case and everyone can contribute different perspectives to the group discussion. If we get stuck, our tutor helps us out and suggests what we could do next. I prefer going to the library to prepare. Here, I can focus and I have quick access to books or journals that help me understand the case. Today, I can also train my stitching skills in the Skills Lab, where you can immediately put into practice what you've learned. After a day like this, I like going to the gym to clear my head and get ready for the next day.
At UM, you meet people from all around the world. Hello guys. Some of them are doing their exchange semester here, and they often say that PBL helps them learn and retain things very easily. I can understand why. It's a very active way of learning because you have to bring your own perspective to real-life cases. You have a lot of freedom to manage your time, your studies, your hobbies, and your work. Of course, that also means a lot of responsibility. Right now, for example, I'm arranging my exchange semester in Madrid. How cool is that? In this group session, we're the managers who have to allocate the resources of a real company. This is how we put into practice what we learned this morning. Studying here means being proactive and learning to plan well. Prioritizing and performing well under stress are great skills that help you develop as a person. But now, it's time to grab a coffee at the Student Service Center.
Right now, we're at the Brightlands Camelot campus. Here we can apply knowledge from lectures and tutorials in a practical setting. This helps us understand what we have learned and further develop our lab skills. Today, we're determining the amounts of cholesterol in various products. What I really like is the project periods at the end of each semester, where we complete a full research project. That includes planning, collecting data, analyzing, and presenting the findings. That way we learn how research works and we're able to see what it's like to be a real scientist. After practicals, I have to write a lab report that also helps me process everything I have learned today. UM has a lot of learning spaces where you can work on your own or with other students. This evening, I'm meeting my friends for a movie night organized by the MSP Study Association.
If you study law, you have to read quite a lot. Not all information is relevant, so you learn how to easily find the information you need to solve your case. In the afternoon, I have to give a presentation, so I like to practice it with a fellow student. Later today, a lawyer is giving a lecture. This will help us better understand the case we're working on. Speaking in front of a group is quite exciting the first time round, but you get used to it quickly. And having to present helps you also to adopt knowledge better. What I like the most is that we sometimes get to enter a plea in front of a real judge. These moot courts are really exciting. Now it's time for drinks with friends. Studying is important, but so is relaxing once in a while.
Helene Widowski, the degree committee here present has assessed the quality of your thesis and your defense. In view of its positive verdict and taken into account your previous qualifications, the degree committee has decided to grant you the degree of doctor. Dr. Wolfs is authorized to confirm upon you this academic distinction in accordance with the Dutch university custom. I invite your supervisor to now take the floor.
Do you promise to work in accordance with the principles of scientific integrity at all times, to be careful and honest, transparent, independent, and responsible?
I promise.
By the authority vested in us by law and in conformity with the decision of the committee here present, I hereby confer you, Helene Widowski, the degree of doctor and grant you all rights attached by the custom and law. As evidence of this, I now present you with the degree certificate signed by the rector, the secretary, and the other members of the committee, and affixed with the official seal of the university. Dear Helene, you made it. Nikki, Tammo, and I would like to congratulate you with your PhD, and we also would like to congratulate your parents, your boyfriend, and all your relatives present here today. It was a tough journey, but with the richtigen Sportgeist hast du es geschafft. Working hard, being well organized, and working as a team member formed the solid basis for a successful PhD project, where you achieved a lot. First, you explored a new field within fetal and neonatal lung biology, where you worked successfully together with neonatologists, pulmonologists, biomedical people, and last but not least, Peter Nichols and Carmen Rijvers from the Department of Pathology in Utrecht and Maastricht. Many new questions emerged, and the project is with Sophie and Tim in good hands. We are really grateful for your excellent role in the large animal work, and I dare to say that the OVA models that have been established by many people, but definitely also by you, are world-leading, and these models will help us to depict important clinical questions in the near future. Finally, with support of the Hubrecht Institute, you have established human and lung organoids of the lungs for humans and sheep that you have used for your own projects, but you also shared the cultures and expertise with other groups within the UM, but also outside our institute. Besides the work in the UM, you had time for many other things. You are a true ambassador of Germany. Good examples are several soccer matches between Germany and the Netherlands, and if the Germans won the game, I received within less than a second after the final whistle a WhatsApp with the national flag, beers and other things, with the question if I saw the game. And you know the answer quite well. The only people that are somewhat disappointed about your promotion work can be found in the Monschau area. Moreover, I have to say you're not single-minded. Your Dutch language skills are excellent, and you're even capable to speak several dialects fluently. Besides this, you had time for sport, you had time for many nice activities with your close friend Louise and with other friends and lab colleagues, and you were always present till the end at parties, lab day outs and other activities. As promoters, you always try to help each PhD candidate to make the essential steps in his or her career. And we shared our last advice with you the past months. You are multi-talented, and please make sure that you use them wise. We wish you all the best and are always there to help when necessary.
dear Dr. Widowski, also on behalf of the Maastricht University, I will congratulate you with this new degree. We, the university, wish you all the best for the future and in your future, maybe academic career. So with that, I will end this ceremony to have a memory, let's say, to this beautiful day. We will take a picture on the stairs of the main hall with the degree committee and the new doctor. And hereby I close this ceremony.
Please put yourself on the picture.